In this video, we are going to talk about experiments. This is our overview slide, which we'll spend about 5 minutes going over before going over each topic in more detail and with more examples. Our first topic is just the definition of experiments. Those are studies where researchers assign treatments to cases. When this assignment includes randomization, it is called a randomized experiment. Randomized experiments are fundamentally important when trying to show a causal connection between two variables. And we need to be careful when we talk about causal connections. We don't want to assume that just because two variables are related or somehow connected, that one variable causes another variable to have certain results. A causal connection, you need to be very careful about concluding. The principles of experiment design is randomized experiments, and they're generally built on 4 principles. 1, controlling. So researchers assign treatments to cases, and they do their best to control any other differences in the group. In this sense, the word controlling, this is not the same use of the word when we say treatment and control groups. So just because we have treatment and control groups, that is not what this principle is talking about. In the main part of our video, we'll do some examples to show how we best control any other differences in the group that might affect our results. The next principle is randomization. Researchers randomize patients into treatment groups to account for variables that cannot be controlled. There's replication. The more cases researchers observe, the more accurately they can estimate the effect of this explanatory variable on the response. In a single study, we replicate by collecting a sufficiently large sample. Additionally, a group of scientists may replicate an entire study to verify an earlier finding. And the fourth principle is that of blocking, which is a little more of an advanced technique, but is often used. Researchers sometimes know or suspect that a variable other than treatment influences the response, and under these circumstances, they may first group individuals based on this variable into blocks and then randomize cases within each block to the treatment group. This strategy is called blocking. Even in a randomized experiment, there's a possibility of having bias, and we want to do as much as we can to reduce bias in human experiment. The first thing we often do is put patients into a blind study, and that's where patients do not know if they are in the treatment or control group. And essentially, that's because the control group is given a placebo. The second thing we do is conduct a double-blind study. So not only do patients not know if they're in the treatment or control group, the doctors that are researchers evaluating the results are also unaware of who is in the treatment and who is in the control group. So double-blind is both the patients and the doctors are unaware. That wraps up our overview, so we're ready to start from the beginning and look at each topic with more detail. So we start with experiments. So studies where researchers assign treatments to cases are called experiments. When this assignment includes randomization, it's called a randomized experiment. And randomized experiments are fundamentally important when trying to show a causal connection between two variables. Remember, in our last video, we talked about explanatory variables and response variables, and the idea is that the explanatory variable will cause a response in the response variables, or they will affect the response. And in experimental, we can show that there's this causal connection between the explanatory variable and the response variable. It's not just an association or some sort of relation that isn't a cause relation. So experiments is a pretty short topic, but we'll go on to principles of experimental design. Randomized experiments are generally built on 4 principles: controlling, randomization, replication, and blocking. So let's look at each of those. The first one is controlling. So researchers assign treatments to cases, and they do their best to control any other differences between the treatment and control group. An example of controlling is when patients are given a drug in pill form. Some patients take the pill with only a sip of water, while others may have it with an entire glass of water. So to control for the effect of water consumption, A doctor may ask all patients to drink a 12-ounce glass of water with the pill. Note, this is a different use of the word control from the use in treatment and control groups. So an experiment that is controlling for different variables is not the same thing as a treatment and control group. You can have a treatment and control group experiment that uses controlling, or you can have an experiment with treatment and control groups that does not use variable controlling. The next principle is randomization. So researchers randomize patients into treatment groups to account for variables that cannot be controlled. And our example is that some patients may be more susceptible to disease than others due to their dietary habits. Randomizing patients in the treatment or control group helps even out such differences and also prevents accidental bias from entering the study. Our next example is replication. So the more cases researchers observe, the more accurately they can estimate the effect of the explanatory variable on the response. So in a single study, we get replication by collecting a sufficiently large sample. So we have many test cases. We're not just basing it on a small anecdotal few cases. Additionally, a group of scientists may replicate an entire study to verify an earlier finding. So often you'll see maybe a US study gets published, and then a different European study might say, yes, we found the same thing. The next principle is blocking. Researchers sometimes know or suspect that variables other than the treatment influence the response. Under these circumstances, they may first group individuals based on this variable into blocks and then randomize cases within each block to the treatment group, and this strategy is called blocking. For our example of blocking, we're going to use the example of a drug used to prevent heart attacks, or that we're going to propose might prevent heart attacks. Even with random sampling, we might get unlucky and put all the patients who are not as healthy, we'll call them high-risk people, into maybe the treatment group. Just by pure dumb luck, we randomly place them into treatment and control groups, but maybe we have a disproportionate amount of high-risk patients that are more likely to have a repeat heart attack into our treatment group. That will bias our results. That's something we don't want. Or similarly, we don't want all our high-risk patients in the control group. So to prevent this, we might first split our patients in the study into low-risk and high-risk blocks, then randomly assign half of the patients from each block to the control and the other half to the treatment group. And this strategy ensures that each treatment group has an equal number of low-risk and high-risk patients. There's a little diagram that goes with this example. This diagram is in your book. So this is our number of patients in our study. It looks like there's 54. And we identify our high-risk patients. They have this gray dot, which I'll just I'm not going to highlight them in yellow, but they're the gray dots over here. Here's 1, 2, 3, 4, 5, 6, 7, and you can see the rest of them. These clear open white dots, those are our low-risk patients. So what we can do is group all our low-risk patients into one group or a block, group all our high-risk patients into a group or a block, and we call it the high-risk patients and the low-risk patients. From the low-risk patients, we split them in half into control and treatment. Similarly, with our high-risk patients, we split them in half in control and treatment. And that way, our control and our treatment group has the same number of high-risk patients, and our control and treatment groups has the same number of low-risk patients. So we're controlling for the risk variable so that we avoid the random sample that puts a lot of high-risk patients into the treatment group or puts them into the control group. We create blocks so that the low-risk patients are in one block, the high-risk patients are in another block. Then we randomly assign patients from each block into treatment or control. Our last topic is about reducing bias in human experiment by having blind studies and double-blind studies. The first thing we'll talk about placebos. So when people are given some medication, it can have a psychological effect one way or another, making the person believe that they'll get better, or maybe they believe for some reason it's a bad thing and they'll believe they get worse. But either way, we want to control for the psychological effect of taking some sort of treatment. And what we do is we give patients in the treatment group the treatment, and then we give patients in the control group a placebo so they don't know they're in the control group. And additionally, then the treatment group doesn't know if they're in the treatment or control group. Everyone gets something that looks like the treatment. And this way, the patients are blind to whether or not they're in the treatment or control group, and we call this a blind study. Furthermore, sometimes we do double blind when not only the patients don't know who's in the treatment or control group, but also the doctors, the researchers evaluating the results are unaware in the treatment and control group, and that eliminates any bias the researchers might have. Certainly, if a researcher works for a drug company, their bias would be to see their drug is effective. And so when they evaluate, they might bring that bias into any evaluation that they do. But if they don't know who's in the treatment or control group, we eliminate that bias. This is a pretty short video, but I guess we'll make up for that in Chapter 2, where some of the topics really do take a lot more time to go through. So Chapter 1's kind of easy. Chapter 2 takes more time. So let's review what we've talked about. We talked about experiments. We said that studies where researchers assign treatments to cases are called experiments, and when the assignment includes randomization, it's called a randomized experiment. Randomized experiments are fundamentally important when trying to show a causal connection between two variables, a cause and effect. Our randomized experiments are generally built on these 4 principles of experimental design. 1, controlling. Researchers assign treatments to cases, and they do their best to control any other differences in the group. Randomization. Researchers randomize patients into treatment groups and control groups to account for variables that cannot be controlled. Replication. The more cases researchers observe, the more accurately they can estimate the effect of the explanatory variable on the response. So in a single study, we replicate by collecting a sufficiently large sample. Additionally, a group of scientists may replicate an entire study to verify an earlier finding. And the last principle is blocking. Researchers sometimes know or suspect that a variable other than treatment influences the response. Under these circumstances, they may first group individuals based on this variable into blocks, and then they randomize cases within each block to the treatment group, and this strategy is called blocking. So even with randomized experiments, we want to reduce bias, and there are 2 things that we do so that people don't perceive their or introduce their bias into the experiment. The first one, in a blind study, we give patients a placebo when they're in the control group, and this way patients do not know if they are in the treatment or control group. The second type of study, the double blind, it's both the patients and the doctors who evaluate the results are unaware of who is in the treatment group and who is in the control group. So that's it for this video. Thank you for watching.